Anyways, so this goes to the P side. So if you see here, this this tRNA, which is bound with this amino acid, it goes here and it binds this. See, this PHE will go to the P and will bind with here. So this tRNA moves here, right? It binds there, and then this amino acid is transferred to the to the ribosome, and then later in the P side, this tRNA goes to the P side, right? And it makes this amino acid be binded to this amino acid. So as it goes on, the chain will get longer and longer and longer. Anyways, just imagine that this PHE was binded to the TRP. So it's now PHE, TRP, and MET. Next, the tRNA that was in the P site will go to here, which is the E site. So after it, releases the amino acid to the chain in order to create the chain of amino acids, it goes here. So now the tRNA is by itself, no more amino acid burdening it. So it goes here, and after it goes here, it just leaves the ribosome. So it goes A and then P and then E and then leaves the ribosome and it goes to the cytoplasm, and that's it. It just boop 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 boop. And the purpose of this is to string the amino acids together in order to create a polypeptide, which is also protein. Yes. So ribosome plus tRNA. What these two things work together and they function together. They help cells. It helps cell translate an mRNA message into a polypeptide. So to combine together, tRNA and mRNA combine together, it helps to make an mRNA message into this polypeptide, which is a chain of amino acids. Now, we want to talk about tRNA. So tRNA is what we call an adapter between mRNA and amino acids. Okay, I'll explain this further. So tRNA, as I said before, it carries this amino acid together with it. So it's like it goes to the amino acid, it attaches with it, and it transports it to the ribosome. And the shape of a tRNA is kind of like an L shape. I don't know why it's called an L shape. It looks more like a T shape to me, but just memorize it as it forms an L shaped feature. So one end, you see this end, but this is square. Anyways, look at this picture. This end, it contains this amino acid, a specific amino acid, while the other, this end here, and more closer, you can just see over here, it contains three nucleotides. For this instance, it's AGG. So these three nucleotides are called anticodons. Now, what is an anticodon? We learned about what a codon is, right? So an anticodon is a triplet sequence of nucleotides on tRNA that is complementary to the corresponding codon of the mRNA. So complicated. So, and more to explain, right? So a codon. Imagine the codon is U, C, and C. Ignore this U. I have no idea why it's here. Anyways, imagine, so this codon is U, C, and C. And in order for it to, like, it binds with things that are complementary, so opposite. So what binds with U? A will bind with C, G, right? So the anticodon will have the complementary, like the opposite one as the codon, in order so that they can bind together, in order so that they can, like that. And so it's a triplet sequence of nucleotides. So there are three nucleotides, as you can see here. Same thing as codon. And anticodons are located on tRNA, while codons are located on mRNA. And the anticodon is completely opposite from what's on the codon, and that actually helps them, like a magnet, to bind to a codon. So think of it like a magnet. For a magnet to attach to each other, it has to have opposite charges, right? Positive and negative will attract. So same thing. The opposite of A is U, so they will cause it to attract. So what an anticodon needs to have are the opposite set of three nucleotides from the codon in order for them to combine and in order